which should be on your screen somewhere south of my chin, right here. And that will come to me as well. And I'll pass those along to Sam. We'll just bake those into the conversation. That logistics now being out of the way, Sam, let's start with what I think is one of the more common questions I get, which is to what extent is CRISPR's perspective on potential approval, launch timing, et cetera, for exa-cel more nuanced than sort of the debate amongst investors? Or do you feel like people have got it within a pretty tight time horizon and the level of uncertainty is pretty low?
Yeah, we're very excited about advancing all the regulatory filings for exa-cel. As Vertex has announced earlier, we've completed the European filings in December. And it's great to see the validation from both the MHRA and the EMA around those filings. And that's all on track there for Europe. I think for the US, we began a rolling submission in November, and we hope to complete it by the end of this quarter. So all on track. So nothing to add there in terms of nuances or qualifiers. I think we're all very busy doing our best. These are pretty intensive efforts around the filing packages, given this is the first CRISPR-based therapy to ever go to regulators. But we feel confident that it should all be done in time, and then we go through the regulatory approval process.
That makes sense to me. Let's pivot to a slightly different topic. As you're talking about different regulatory authorities, one debate that's arisen more in vivo land than in therapies exactly like this, but certainly relevant to your pipeline, is the perceived gap between the expectations to dose first patient in the US with in vivo gene editing tool versus the expectations of European regulators, Asia-Pacific Rim regulators, and also regulators in countries in Oceania, New Zealand, Australia, et cetera. How wide is that gap in terms of the expected pool of data to dose your one's first patient for gene editing? We'll start with just that. How wide is that gap?
It's hard to genericize. I think it's program by program. I think depending on how severe the disease is, what the unmet need is, you could have differences in how the regulators set standards. By and large, at 20,000-foot level, there's not that much of a difference. I mean, I think it may manifest itself in terms of a few months here and there, but what I would say is, there may be some differences in the length of tox studies or the size of preclinical tox studies, some of the assessments you need to do for off target, et cetera, but it's not dramatic. I mean, we went through this with our ex vivo therapies back in 2017 timeframe. We started with the CTA in Europe and dosed our first patient in Europe, and then we filed our IND in the US. We did go on hold for our sickle cell therapy at the time, which caused a lot of the same questions and sort of excitement around the difference in regulatory standards. But it turned out that it was one of those where it just takes a little bit of time to standardize and fully ensure harmonization of requirements, not just across the ocean, but across different teams at the agency. So I wouldn't read too much into it at this point. I think at this point we do have to do a little more work for IND than we have to do for certain other approvals, but it's not dramatic.
That makes sense to me. So as you talk about sort of harmonization of standards and sort of narrowing that, have we reached harmonization in ex vivo world? And is this just sort of like a nuanced debate in terms of the in vivo side of things, where theoretically there's still somehow potential gonadal exposure? Is that really the driver of it?
Yeah, so I think for ex vivo, you have to take it part by part. I think there's the non-clinical, there's the CMC, and then the clinical standards, right? The clinical standards, I think really depend on the indication. It's very hard to say that there's standardization. I think we've seen a number of indications where different regulatory agencies have taken a different view as to what's required for a filing package. Take one of the autoimmune drugs, for instance, that's is approved in Europe but not in the US, and there was a refusal to file, for instance, 2 years ago for one of these drugs without naming exactly what that is. But I think for the more important question is the CMC package and the non-clinical. I think if there were widely varying standards on CMC, it would cause a lot more work and a lot more expenses to do it all. But I think there is actually a lot of convergence on what's required on the CMC packages. And similarly, I think on the non-clinical aspect, there is convergence on or there's, I would say, things are getting closer on what's required from preclinical tox packages or assays and everything else. I do think that over time you're going to see the same convergence in vivo therapies, but there's just that higher scrutiny given that you want to rule out any chance of editing in organs that you're not going to want to edit in. And let's say if you're targeting the liver, you want to see that most of the editing is happening in the liver and not much else in any of the organ systems. So again, all this will get worked out. There are a lot of forums for industry and the agency to work together, and there's support from NIH and other bodies here as well. So something that I don't think is going to be a major concern for the field in the long run.
Let's narrow down a more specific, split to your program, probably Vertex, in hemoglobinopathies. How do you, are there specific lessons you're going to be drawing or factors you're going to be following in your competitor Bluebird's experience? A different technology, to be fair, but not entirely different population. As you form your own commercial strategy, and are there things that you've learned, that you and your partners have learned from their experience in Europe that are influencing your entry into that market?
Yeah, I think Vertex is leading our commercial efforts, and they will provide much more color and commentary around specific efforts in the U.S. versus ex-U.S. What I'll say is I'm not entirely sure that there's much to take away from the Bluebird experience in Germany. I've generally found that some of these European payer systems will come to the table and negotiate the appropriate price for drugs as long as there's support from ICER and other bodies, but also that there's significant value demonstration. What may have happened is a one-off situation that may not actually be much of a telltale sign for what's going to happen to exa-cel in Europe. So we're not leaning too heavily on that experience. I think what is encouraging is as Bluebird is launched in thalassemia in the U.S., which is a much smaller addressable population, it does seem like there's reasonable interest in the therapy, and it'll take some time for them to enroll, get the patients on to manufacture their product and dose them. But it does show you that there's patient demand, which is what I've been saying all along. I do think the street is undervaluing exa-cel or therapies in this category, because the patient demand we see and the excitement we see in our clinical trials tells you that there are a lot of patients who are just fed up of living with this disease, and they want to take a chance on a therapy that could cure them, especially after seeing some of the stories that we saw in NPR and other outlets. So I do think the demand equation is going to be strong.
So we talk a little bit about the demand equation. I think that calls to mind a very different market, but another one with a complex manufactured product, which is Bristol's experience in cell therapy world, where they ran up on a manufacturing supply limitation. And it's easy to chuckle and call that a high class problem. But it is a problem because patients who need drug are getting drug. Mm-hmm. There isn't the same ticking clock in sickle cell of potentially progressing on a tumor, but you could have a crisis or stroke or end organ damage. So, so in some ways there is a clock. How do you think about you and your partner scaling the manufacturing capacity you need, having it in the right places geographically? Like, what is the plan? How much flexibility needs to be built into that plan for over/under supply within defined geographical areas?
Yeah, it's a really important question. And, I think what I'll say is both Vertex and us have had the same philosophy, which is we want to make sure there's drug supply for every patient that wants to get the drug and deserves it. And, part of that is how we scale up manufacturing. Fortunately, given the strong balance sheets on both sides, we've actually scaled up manufacturing to a point where we don't think we're going to be drug limited, supply limited. The second thing is we also create some redundancy across the Atlantic so that, we don't have to fly the samples out too far, and we can turn around manufacturing faster in the same region. Now, in the early days, you do have to sequence them because, when you harvest the cells from the patient, even if you have, 10 patients day one, you have to harvest the cells, send them to manufacturing, and there's a normalcy that comes to bear after a few months in terms of the manufacturing schedule. But we're also preparing to ensure capacity for the upfront bolus if there is one. So all these things that we're taking into consideration, and we're being, relatively more aggressive about it, just to make sure that we don't run into a supply issue.
Right. while we're digging in on some of this nuance, how should we think about the applicability of infrastructure built around this partnership and how it might apply elsewhere within your partnered assets with Vertex and also elsewhere in your wholly owned pipeline? Or is it unique enough and the manufacturing process sufficiently unique that that's the wrong way to think about it?
Well, I think we've had the benefit of being able to work with Vertex, and they have a large setup that we were able to learn from. One of the key reasons for us to go into immuno-oncology with cell therapies was the parlay that we had from our hemoglobinopathies program in terms of competencies. We learned how to handle cells, how to do the release testing, how to do the quick turnaround, and that came in very handy as we moved not just the first gen programs in immuno-oncology, but also the second gen programs. And so we have been able to expand on that competency. I'm going to almost call it a core competency for us now, which is cell handling. For instance, that came from our lead program. Second, I think for our diabetes program now, with the acquisition of BioSite by Vertex, we are partnered with them 50-50 on our VCTX 211 program. And there it's a very different disease, very different situation from a manufacturing standpoint. But I think from a decision-making standpoint, I think having worked with them closely on exocell, we're able to carry over some of the same operating constructs or the operating agility into that program in diabetes, where we're dosing patients. So there is definitely benefit as we've tried to expand our portfolio from having a successful lead program. And by the way, as we get to, hopefully to BLA filing for our immuno-oncology products, the experience of having gone through exocell is going to be tremendously helpful because, the same work we've done, for instance, in CMC to qualify our enzymes, our process of making guides, et cetera, all that ports into additional BLAs that we may file.
That makes sense to me. As you brought up, let's move over to the oncology side of the platform for a little bit. I don't want that to get ignored, and I know that that sometimes gets lost in all of the nuance and focus on the scale and size of the sickle and beta thal opportunity. So I think broadly speaking, the discussion has been that an expectation that CTX 110, will be dosing a registrational study sometime early this year. That being the case, on what time horizon is it reasonable for us to expect kind of initial data from that study?
Yeah, I think CTX 110 is already in the registrational trial. And I think, when it's in a registrational trial, there's defined points at which you cut the data. You don't want to compromise the data set by looking at it too often or cutting the data based on when we want to disclose it. So there's an interim analysis baked in. And depending on enrollment speed and how quickly, how the patients do on the trial, we'll hit those interim analysis points where that's when we would cut the data and disclose the data. So 110, hopefully we get to that point this year, but we're advancing the trial rapidly. On 130, I think we have a meeting with the agency based on our RMAT designation to clarify what a registrational trial might look like for CTX 130 and T-cell lymphomas. This is a rare disease which has no other therapies. So what is the bar really? How do we think about it? Trial design. Even if it's a single arm trial, what are the endpoints we'd be looking for approval? That's an important discussion that we have upcoming with the agency that would dictate what that registrational trial looks like. And then again, we'll have interim analysis points baked into that trial as we start that trial, start the registrational part of that trial. And finally, I would say for our next gen programs, 112 and 131, I think we're in the clinic, and I think we'll have data this year. The question is, at what point we disclose the data, but there's a lot of excitement around those two programs. Not just from investors, but from pharma companies and investigators. I think the fact that we saw a response with CTX 130 in solid tumor in RCC, complete response nonetheless, and then the additional data that others saw with CD70 CAR T and RCC gives us great confidence that a more potent CAR T in RCC could be transformational. Now, we have to dose patients and make sure it's safe, and that actually bears out in terms of the data, but there's quite a bit of excitement in the field around CTX 131 and CTX 112 as well.
So while we're on this topic, I think it makes sense to talk a little bit about the next-gen constructs that get a little bit less attention, which is understandable for investors who think about things a little more like almost a DCF timeline. So what happens tomorrow is more important than what happens a year from now, but perhaps not the way one should think about operating a company. Let's talk about the timeline on when we're going to get incremental detail for 112 or 131. And then we'll start talking about, your perspective on some of the additional edits and, like exactly where we're looking to increase potency versus current generation proteins.
Yeah, I mean, it's not too far in the distant future. The two edits that we incorporated with our CTX 112, 131 edits programs are regnase 1, which is a ribosomal binding protein and a transcriptional regulator of cytokines and pro-inflammatory environment, and TGF-beta-R2, which is a well-known target for making these CAR Ts less exhaustion prone in the tumor microenvironment. And regnase 1 came out of a large empirical screen and also was bore out in one of the partnerships we had. I think it's going to be a very important target in immuno-oncology. It hasn't been one because it's not extracellular and easily targetable, but intracellular, as an intracellular target, is very important. And so we are, what we're looking for is the expansion kinetics early on. Within the next 6 months, we'll know how these cells expand in patients and whether it's much higher expansion relative to the first-gen products. Now, we haven't decided when we're going to disclose the data, and we want to get a bigger clinical data set before we disclose it. But the telltale signs are there. If you see much greater expansion of the cells, if you see that the cells are much more central memory phenotype or more naive phenotype, let's say at day 10, if you see that the cells are not exhausted by day 12, day 14, all those are very important markers that would prognosticate how the trial might work out. And so we're quite excited to see that over the course of the next 6 months. Whether how we disclose it is something that we'll guide to middle of the year.
That makes sense to me. We talked a little bit about the impact of those edits and potential benefit. One of the questions that I get from investors a lot is, how might that increased potency sort of declare itself in clinical studies? Like what hints should we be looking for in sort of initial readout versus sort of a longer term follow-up readout? Do we need to see multiple doses to judge a dose response, the degree of benefit? Like what are the metrics that investors should be looking at in the early first couple of readouts for these assets?
Yeah, absolutely. I think let's take the example of 131 in RCC. The first thing we're looking for is the expansion kinetics, as I talked about earlier. Are we seeing favorable expansion kinetics relative to first gen? Second, of course, is to say, are we seeing responses, and are we seeing responses at doses that are much lower than what we saw with the first gen, right? If we saw, if you started seeing responses at dose level 1 or 2, that indicates that your data might get better because there is a dose response with these CAR Ts. And we saw some of that, at the last ASH, there were some academic posters or work done by academics around CAR Ts armored with IL-18, for instance. And, instead of seeing responses at 300 million CAR T cells, we start seeing responses at 30 million CAR T cells because the cells are so potent, right? So, these drugs declare themselves early if their potency is higher, assuming we can stay within the safety index. So that's what we'd be looking for, and it would inform how we think of our own investments, because if we start seeing those signs, we're going to go pedal to the metal on other IO constructs, other targets with our allogeneic CAR T platform, especially now that we have our own manufacturing and we can turn it around much faster.
That makes sense to me. A few minutes left. I want to pivot to the in vivo side of the platform. One of the questions that we get is, given that there's different companies looking at very different indications, is at what point is the indication too broad market? Especially this is in the context of CV disease, et cetera, for Crispr to want to build out its own commercial infrastructure. Is there like a threshold where you think, okay, this is more appropriate for partnering, or is that driven by indications? Is it driven by size of the required sales force? Like, how do you think about indications that belong in-house in vivo and indications that really belong in someone else's hands commercial?
Yeah, no, I think it's for us, it's more a question. It's not just about commercially and the commercial infrastructure required. It's what are the development costs? I think if something's going to take a 5,000 patient trial, it's pretty expensive for a small biotech, right? So I think that kind of informs a little bit our choice of indications. We're going with ANGPTL3 first, LPA next, and then PCSK9. With ANGPTL3, there's only one approved drug, which is Evkeeza. And we do think that you can actually do a relatively small trial in populations like, high triglyceride population, which is not, thousands of patients, it's hundreds of patients at best, or maybe a couple hundred patients, and get approval, which is something that we can completely do ourselves and potentially even consider launching by ourselves. Obviously, we'll make that decision at the right time. But the true value creation is going to be between now and HPOC, and having a path towards BLA from there. So that's something where we think about quite carefully, because that equation may be different for PCSK9. It may be a little more expensive to develop PCSK9 all the way, and I'm not sure that's something that we want to take on through the entire course of development. LPA is a very interesting target. I think the more we look at the emerging data, and obviously there's big data readouts coming from Novartis and then Amgen as well on LPA, and that could be a very interesting target that we want to retain full control of at this point as we see what other data is coming in as we continue to prosecute our own program. But what you're asking are very important questions, and questions that we think about all the time. But our key metric is, a return on invested capital, and making sure that we do see a 3-4x return on enterprise value based on the invested capital that we see in the near term. And then we can always make that decision around partnering later on beyond that point of extracting value.
That makes sense to me. We're coming to the very end of our time here, but one of the other questions that comes up quite frequently is around targeting for in vivo approaches extrahepatically. Obviously, there's a debate, and a couple of different companies have taken swings at a few different technologies, pulmonary, et cetera, muscle, bone marrow. How do you think about the landscape of novel targeting technologies to endeavor to deliver a construct to gene edit other tissue types? Are there specific technologies out there that you're looking at that you're impressed by? Are we still in the early days and no one's really developed proper proof of concept?
Yeah, this is actually a big focus for us. I think for neuromuscular indications, it's turning out that viruses are a better vehicle than LNPs. And so we focused on improving the virus capsids, one with our partner company Capsida, and another with our collaboration with Vertex in DMD and DM1. And that's the route we'll take for those indications. But LNPs are turning out to be quite useful for other organ systems. We can think about the eye, you can think about the hematopoietic system. You could target LNPs specifically to those organs. Obviously, all these things are easy to conceptualize, hard to do, because if you start the conversation off with all the regulatory hurdles around, just LNP delivery to the liver and the constraints around it to get an IND, I think it's that much harder with other organ systems to ensure that you have specific delivery to those organ systems and not anywhere else. But that, all that's moving much faster than we all anticipated, I think, 3-4 years ago. And I think LNP technology, especially armored LNP with biological conjugates around certain targets, are gaining a lot of traction.
Great. That is very helpful. We're out at the end of our time message here. Sam, thank you so much for joining us. I'm certain that a lot of these questions are going to be revisited over and over the next couple of years as you and your partner Vertex enter the commercial realm together, and as well as a lot of these novel technologies expand the reach of CRISPR gene editing. I'm looking forward to those conversations.
Terrific. Thank you, Marty.
Pleasure as always.